just a few basic facts about vaccine design. So neutralizing antibodies, as I said, these are important, and they are directed against the major viral surface protein. So viruses, and typically I'm largely talking about enveloped viruses, which have a cell, which have a membrane on their surface. On their surface they have proteins, and those proteins bind to receptors on target cells, and that's the first step of infection. So the SARS-CoV-2 virus, which is a causative agent of COVID-19, it binds to a molecule called ACE2, which is present on many cells. So the antibodies that now, the first thing that the immune system sees is the outside of the virus, right? And so that's what you generate antibodies to. And so these antibodies, if they can block the binding of the virus to the receptor, then they'll prevent entry of the virus into cells. So that's obviously important. Now, as I mentioned earlier, these viral surface proteins are often metastable, so they display a large number of conformations. Many of them are irrelevant. And now, in order for the virus to enter the cell, especially if the virus has a membrane, there's a big barrier because the cell also has a membrane, and the virus needs to get past the cellular membrane, and the proteins on its surface, like all proteins, are somewhat charged, and so for those charges to go through the membrane is not a straightforward process. And also, because now the viral membrane and the cellular membrane, or if it gets taken up into endosomes, the endosomal membrane, they both act as barriers, because ultimately the virus has to enter the cytoplasm or the nucleus of the cell if it's going to replicate. So in order for that to happen, you need a process called membrane fusion to happen. So the viral membrane and the cellular membrane can either be the plasma membrane on the surface of the cell, or it can be an interior, like if the virus is taken up to endosomes, it would be the endosomal membrane. Now these membranes need to fuse with each other so that the viral contents go into the cells, and that process requires energy because you have two negatively charged surfaces which have to mix with each other. So that process of membrane fusion requires energy to happen, and that energy in many viruses happens. The source of that energy is not ATP hydrolysis, because you know there's no ATP anywhere. It's all the outside of the virus. That happens because the virus, during the process of fusion, the surface protein on the virus transitions into a much lower energy conformation called the post-fusion conformation. Okay? And the energy for that process is what drives fusion. And so because the viral surface proteins are in a metastable conformation, that is, the native state is not the most stable state, many times the antibodies that are generated will be generated against the post-fusion conformation, because they are viral decoys in which the surface protein is in the post-fusion conformation, and those are useless for a vaccine, because the vaccine has to recognize the native metastable state of the fusion protein. And besides neutralizing antibodies, T-cell immunity, which is cellular immunity, also contributes to protection. And what's the function of cellular immunity is to recognize infected cells, right, and target them for destruction by the CD8 T cells and other cells of the immune system. But we are not going to discuss cellular immunity here. Now, as some of you are probably aware, DNA sequencing costs have been steadily going down and down and down, right? So the human genome is about 6 billion base pairs long, and currently the cost of sequencing a genome is just a few $100s. Okay? And you know, most viruses are much, much smaller than this. And you can see, so Moore's law, which is what you have in semiconductors, is a reduction in costs of manufacture, and it's been so those prices have steadily been going down, which is why we have better and better electronic devices. They've now somewhat saturated because of they've reached a certain dimension of atomic dimensions beyond which things can't be packed in too much further. But now if you look at sequencing costs, right, This is the period when we had Sanger sequencing. Everybody knows what Sanger sequencing is? Yes? Okay. So now what the development that actually happened in the mid 2000s is that there was the development of what's called next generation sequencing. Now the big difference between Sanger sequencing and next generation sequencing is that in Sanger sequencing we are sequencing a collection of molecules, right? So you have a plasmid, and in solution there'll be, you know, millions of copies of that plasmid molecule, and they'll all be identical, and that's what you are measuring. What next generation sequencing allows us to do is to measure the sequences of individual molecules in a mixture, okay, and get all those collection. So if you had, you know, millions of different plasmids all in one test tube, what next generation sequencing would allow you to do was to get the sequence of each and every plasmid in that complicated mixture without having to separate them. Okay, so that's the technology which we won't really get into. So how can we use these advances to make better vaccines? Now there is, are people here familiar with what is site-directed mutagenesis? Yes? Okay. So site-directed mutagenesis, what it allows you to do is, if you have a sequence of DNA, it allows you to change one or more base pairs specifically, okay, in that sequence. So a few years, about, you know, 10 or 15 years ago, we and other people took advantages of these developments in next generation sequencing methodologies to create mutant libraries. And the essential way this is done is you create a mutant library. I won't say how we do that. Now you put that mutant library through a screen, okay. So supposing you make a bunch of mutants of a gene that is involved in antibiotic resistance, right? Now you want to know which of those amino acids is contributing to the ability of the protein to produce antibiotic resistance in a bacterium. So what you can do is you can create a library in which you are mutating every amino acid in the protein to every other amino acid, and this is done in parallel, so that you have, you know, tens of thousands of different mutants in your pool. So at each position, if you have 20 possible amino acid substitutions, okay, if you have 200 amino acids in your protein, how many single mutants would that correspond to? If you have 200 positions and each position can have 20 substitutions, how many are there totally? Nothing complicated here, right? What's the total population of single site mutants that you can have over 200 amino acid protein? Huh? No, it's not 20 to the power 200. It's 200 into 20, right? It's a linear problem. It's not a combinatorial problem. So if you're talking about single site mutants, there are 4,000 possible single site mutants that you can have. The moment you have multi-site mutants, right? If you have, supposing we wanted to know that what are the total number of two position mutants, right? Where any two positions can be randomized. How many mutations would we have then? Huh? No. So at a given position we can have 20 amino acids, right? Now we have 200 positions, out of which 2 are mutated. So how many different combinations can we have? Huh? Yeah, 200 C2. So at each position, right, we have at each position we can have 20 different choices, right? So the number of possible choices that you have will be using the combinatorial formula. So that number, okay, is still, it's not millions, right? It's of the order of tens of thousands, okay. So now with the possibility to do next generation sequencing, it's straightforward to be able to sequence this many mutants in a pool, because the human genome has much more than the number of, for example, 2-position mutants. But even if we are interested only in single position mutants, right, what this methodology allows us to do is to take all this collection of single point mutants and then find out, if we have a functional screen, which ones of them are active. So we'll start with single mutants because that's the easiest to do, right? So if we have a collection of single mutants of this hypothetical protein, we can see initially how many single mutants are there in the pool. Then we put it through, let's say, an antibiotic selection, for example, and we can sequence the plasmids that are there, just the gene in the plasmids from that collection of bacteria, and that will tell us how many mutants have survived the screen, how many mutants are functional. And by looking at the difference, we will know how many are non-functional. Okay? So in a single experiment, we can get all this information. So we've used it over the years to do many things. During the COVID pandemic, we were trying to apply this to stabilize protein. So we all know there are plenty of vaccines available for COVID, to fight COVID-19, but all these vaccines require refrigeration and some of them require frozen storage. So the worldwide vaccine coverage is quite uniform. So if you look at a map, you'll see that the bulk of the vaccine doses during the pandemic were distributed in the US and Europe and Japan, well-developed countries. And there were many parts of the world that had hardly any vaccination. And one of the reasons for this, besides the fact that there were no vaccine manufacturers in many countries, is that they were not able to have the infrastructure, the cold chain infrastructure in order to be able to transport and keep vaccines cold. So for many situations, it's important, even though there are some existing vaccines, to have vaccines that don't require a cold chain. You're all probably familiar, there have been a lot of advances driven by artificial intelligence and machine learning, which you encounter, in your day-to-day life with ChatGPT and so on. And the same kinds of principles are actually being applied to prediction of protein structures. So large language models, there's been a lot of advancement in applying large language models to predict protein structures and applying machine learning to predict protein structures. So despite all this wealth of ability, the prediction of stabilizing mutations remains challenging, and the reason for that is all these advances are based on having a large amount of data available. Okay? And we don't really have huge amounts of data where we know that if you change one amino acid to another, what is the energetic effect of that in a protein or in thousands of or millions of proteins. Okay? That kind of data is not available. Now it's easier to generate that kind of data, but it's not yet available. So this still remains challenging. So the way we went about doing this is, one of the things we use in my lab is a method called yeast surface display, which was developed at MIT. And the principle of yeast surface display is that you display some protein which you can clone, any protein of interest, on the yeast surface, and you can detect how much of that protein is produced by an epitope tag. So it's a small sequence which is recognized by an antibody. And if you have a ligand which has another epitope tag, then you can detect the binding of that ligand to the yeast expressed protein. So instead of having a single protein expressed on the surface, you can have a library of proteins. So where each yeast cell will display a different protein. Now, so there are many display methods. There's yeast display, there's phage display, mammalian cell surface display, and so on and so forth. And they're very good if you want to identify mutations that improve the binding to your target of interest. Okay? Because you can screen lots of things by making a library. But now if you want to make a more stable protein, it's not directly good for that, because as long as your original protein is stable, it's going to bind the ligand very well, and even if you have a more stable protein, it's not going to result in better binding. Okay. So now um, the way we got around this problem is that after we made our library in which every amino acid was mutated to every other amino acid at each position, we introduced a destabilizing mutation into the library. So what the destabilizing mutation does is it prevents the protein from binding because it becomes unfolded. And now when you introduce this into the context of all other single site mutations, you have a two-site mutation library in which every member has this, and then you have all these others. So that combinatorial problem then doesn't happen because everything is in the context of this one destabilizing mutation, and we can then select for mutations which suppress the effect of the destabilizing mutation, and they actually often turn out to be stabilizing. So that's in fact what we did. Okay. So we got these suppressors, and then once you introduce the suppressors back into the wild type gene, those are often stabilizing. Okay. So the way we do this is we make libraries, display them on the yeast surface, and then we sort the different parts of the library, and we subjecting them to deep sequencing. So here, as we go further and further along this FACS histogram, these are better binders, these are less good binders, so on and so forth. And so we are able to, from a single experiment like this, we are able to find out how well each mutant in our library is binding. And then what you get at the end of all this is a heat map. So these are positions along the amino acid sequence, and these are each of the 20 substitutions. The white places are where we couldn't get data. And so, we don't have time to discuss all this, but the red ones are ones which are binding better than our destabilized protein, right? And so from those, so the red and the yellowish ones are all the mutants which are likely to be stabilizing. So in a single experiment like this, you can get a huge collection of potential stabilizing mutations, and then you can actually verify them by putting them back by site-directed mutagenesis into the individual protein and measuring the stability. And by combining two or more such mutations, you can get very large increases in the stability of the protein. So about 15 to 20 degrees with just a couple of mutations that you isolate by this kind of methodology. Okay. So next, we wanted to show that this worked for lots of different proteins. The protein we had initially taken was a small bacterial toxin. We applied it to a surface protein from HIV GP120 to the receptor binding domain of the COVID spike protein, and then to a bunch of other proteins from other sabicoviruses. And in black is the protein that we got through our approach, and in red is the wild type protein. And you can see in each case we were able to get large increases in stabilization and also in thermal tolerance. So if we took the wild type protein and exposed it to elevated temperatures, then you can see there was a complete loss of activity. But when we took our stabilized proteins, then even up to 60 degrees, they retained their thermal properties, and so on and so forth for a bunch of other proteins. So we could use this kind of approach in a robust way to enhance thermostability. So then next we turn to, this is the spike protein of the SARS-CoV-2 virus. This part is the receptor binding domain in red, and that binds to the ACE2 receptor on human cells. So this can be well expressed in multiple different hosts, and we showed early in the pandemic that this protein is very thermotolerant. So in other words, if you freeze dry the protein, remove all the water, you can heat it up to boiling point temperatures, and you can bring it down, and the protein still remains folded, so it folds reversibly. But when you use it as a vaccine, it doesn't generate enough antibodies. So we wanted to see through stabilizing the protein through our approach, whether we could get an improved vaccine candidate. And so we, as I said, we showed that with a few mutations we could stabilize the protein. And then we injected it into mice and we looked at the neutralization titer. So the neutralizing titer is once you immunize mice, you take out the sera from the immunized mice and you see whether the sera are able to prevent viruses from entering cells, right? And the dilution of the sera that you can make, the maximum dilution that you can make, so that you still get at least 50% cell entry, that's called the neutralization titer. So you can see for the wild type protein, the sera we could dilute about 1,000-fold and we could still get neutralization, whereas for the mutant protein, it was about 10 to the 5-fold. So just with a few mutations, we had increased the neutralizing potency about 100-fold. And when we looked, so in December of 2021, the Omicron virus came into the human population, and essentially all the wild type vaccines couldn't generate sera that would neutralize the Omicron virus. But when we took the sera using the stabilized protein, we still got very substantial neutralization of that virus. So next we wanted to further improve the properties of the protein. So it turns out the spike protein has a very conserved subunit called the S2 subunit. And so we fused this receptor binding domain to the S2 subunit, and we showed that if we challenge the mice with the COVID virus, you can see the unimmunized mice rapidly lost weight and died, and the immunized mice retained weight and they were perfectly fine. And again, because of shortage of time, I won't spend a lot of time on this, but the major, the protein that's used in all COVID vaccines is the full spike protein. Okay, that's the whole surface protein, and ours was something that was much smaller than that. And again, to cut a long story short, we showed that our protein was better than the spike protein that is used in basically all vaccines worldwide, including the mRNA vaccines encode for the full-length spike protein. So in green, you can see that when we challenged hamsters which had been immunized with the full-length spike protein, they lost some weight and they didn't fully recover weight at after a few days. But with our candidate, they recovered weight quite well. And these are histopathology slides from the unimmunized and challenged animals. You can see that all the lung spaces have shrunk. This is with the spike immunized animals, and this is with our formulation. So it's a little bit better. Okay. So next, in the next 5 minutes, 5 to 10 minutes, what I'll do is, we wanted to see if we could apply our methods to map the epitopes that were targeted by the vaccine-induced sera or through viral infection. So when you have either viral infection or vaccination, there are a bunch of antibodies generated, and these antibodies, which are different in different people, they bind to different regions of the surface protein. So in order, a very important criteria for vaccine development is you must generate neutralizing antibodies, which can be assayed for, but importantly, we would like to know where all on the surface protein are the antibodies binding. And this is complicated because it's not like you have two distinct molecules and you just have to find out exactly where they interact. Here you have one molecule, which is the spike protein or the RBD or whatever which is in your vaccine, and then you have all the different antibodies that are generated by immunization, and they will bind to different places on the surface of that protein. And you would like to find out where all are these antibodies binding and how does it differ depending on the vaccine, depending on the person, etc., etc. Okay. And so we used an approach again using yeast surface display, and the idea here was that we now put charged amino acids all over the surface of the protein. And the idea is that if you have an amino acid that is not charged and you replace it by a charged amino acid, if that amino acid, the original amino acid, were part of the epitope, that is, it were involved in binding the antibodies, then when you replace it with a charged amino acid, you will get a loss of binding. Whereas if it were not part of the epitope, then you wouldn't see any kind of effect. So what we did was we made this library at which all the surface positions were replaced by a charged amino acid. We chose a negative charge aspartic acid, except where the wild type of amino acid was negatively charged, where we put a positive amino acid. So we made this library and then incubated for a given serum. We would incubate the serum with the library and detect which antibodies were bound. Right? So these are the bound antibodies, these are the unbound antibodies. And then we would deep sequence the library, both of these populations, so we could figure out which mutants were not binding to the antibodies present in the serum. And those mutants would be the epitopes targeted by the antibody, because after mutation they have stopped binding. Now, in order to figure out the mutants a little bit easily, what we did was each mutant was fused with what is called a barcode. So you all know what barcodes are, right? They're specific sequences which are associated, for example, with a product which you buy in a store. So here these are molecular barcodes which were outside the gene. So we know that if we have the sequence ATGC over here, this corresponds to this substitution at this position. If you have a different barcode, it corresponds to another substitution. So now the advantage of this is that when you do the deep sequencing, you don't need to sequence the whole gene to figure out where the mutation has happened. All you need to do is to sequence the barcodes. Okay? So we didn't invent this methodology. It was invented quite a long time ago by other researchers at the University of Washington. But this allows you to do this kind of analysis very quickly. So I won't go through the data analysis, but essentially this is the kind of data you get. So these are different sera from different individuals that were collected by our collaborators, and these are the positions in each serum where binding was affected by putting the amino acid substitution, right? So in this single experiment, right, so each of these corresponded to one yeast display scan, where we had our library, incubated it with the sera, found out which mutants were binding, which mutants lost binding. And we could do this for multiple sera, and then pool all those data together, and this is what we get. Okay? So these are positions, and you can see that they're slightly different. They vary a bit from individual to individual, which is what you expect. And some of these residues are already known to be a part of antibody epitopes, but some were actually not known. And there was a recently discovered epitope called a class 5 epitope, which is away from the epitope from the ACE2 binding site. And what we found was that the people were individuals that we looked at were raising antibodies to this newly discovered epitope. And the reason this is both interesting and important is that these sera were collected around, so these people were vaccinated in about, early in the pandemic, or so just before the Delta wave or around the Delta wave. And then the sera were collected around the end of December and the beginning of January, and that was after the Omicron wave had just started. And the interesting prediction from these experiments was, because these residues were conserved, these sera should be able to neutralize viruses that came up much later in the pandemic. And in fact, that turned out to be true. So as I said, most of these sera were targeting this so-called class 5 epitope that we looked at, and that had not been found in studies with populations abroad which were using mRNA vaccines. And to confirm that, we showed that these sera could also neutralize the very recent XBB 1.5 virus, because those residues had not been mutated in those viruses, in that virus. Even though the actual infection and the sera were taken from these individuals well before the XBB virus appeared on the scene. So the important conclusion from this is that the immunity that was generated by these breakthrough infections and vaccinations in India early in the pandemic remain effective against the recent variants of concern. And so this behavior is, this kind of properties are a little bit different than what has been seen in highly vaccinated US population. So just to conclude here, so we've used saturation mutagenesis coupled to yeast surface display, and we can show that this is a good method of identifying stabilizing mutations, and currently it's superior to computational methods for doing the same thing. We got a very good vaccine candidate, but nobody's interested in COVID vaccines right now. So we are combining this with a flu formulation, and we hope to put it into clinical trials soon. And the same way we can stabilize proteins, virtually any other protein of interest. And then we were able to use related methodology for rapid epitope mapping in polyclonal sera. And the libraries that we use here are much smaller than those where we have complete mutational scans, where you have thousands of mutations in each library. Okay. So these are the people who did the epitope mapping work, and this was done by the sera were provided, and some of the experiments were done in Dr. Arindam's lab at IIT Kharagpur, and the sera were provided by many of his clinical collaborators, and Kokab is the person in my own lab who made the mutational library and did all the deep mutational scanning for these. Now for the rest of the work, there are a lot of people who contributed. So these are all various people in my lab who worked very hard during around the time of the COVID pandemic and just before, to develop some of this methodology. And Professor Shashank Tripathi and Somnath Datta also were involved in some parts of the work. All the pseudoviral assays were done by my collaborator at IMTech, and these are people at the Vaccine Formulation Institute in Switzerland who supplied us with adjuvant to do the immunizations. And we got transgenic mice from NCBS, and Minvax is a vaccine startup that I co-founded a few years ago, and they did all the serology studies, and they were invaluable in getting this work done. So that's it, and I will conclude over here.
Thank you, sir. If there are any questions.
Great talk, sir. To generalize, what is it about a protein that makes it thermostable or not? Like you said, the vaccine you developed was stable at 37 degrees.
Yeah.
We have read in a class 12 of enzyme stable at temperatures high at 70 or 80.
Correct.
So what about the protein structure determines at what temperature will it degrade?
Right, so that, there's a lot of interest. It's not a fully solved problem. So one thing is known, that in proteins from thermophiles and hypothermophiles, there are more salt bridges on the surface of the protein. So those cause some degree of stabilization. But as I say, we don't have enough information right now to, with high accuracy, rationally design such mutations. So we can put salt bridges on the surface of a protein. Many times they don't do anything, they don't stabilize. We can put disulfides in a protein. Disulfides also are stabilizing interactions, but the success rate of all of these is not that high. So that's why we took a somewhat unbiased approach, where we just made a whole bunch of mutations, because now the technology allows us to do that, and we just selected for things which could stabilize. somewhat, well, we didn't go completely, I think it's easy to predict which mutations will destabilize the protein, right? So the inside of the protein is pretty hydrophobic. If you put a charged amino acid there, it's obviously energetically unfavorable. So you can eliminate many mutations in that way. But to figure out which are the stabilizing mutations is more difficult. There are many mutations that you can make and which you don't know whether they'll be stabilizing or not, and that's why you need to screen them. So as more and more data becomes available, then you'll have to make fewer and fewer mutations. But right now, you still have to make a bunch of mutations.
Okay, Professor, I had a question.
Yeah.
I wanted to know about other methods of stabilization, like crystallization. Do you think that there is challenges with that, or why isn't that more explored?
No, so crystallization is a method for getting structures of molecules.
Okay, because I see companies like Merck sending pembrolizumab to space just to crystallize them because they say that they can reduce the cold chain requirements and it's a more stable form of the drug.
Well, you know, the you mean the protein is administered in crystalline form?
I believe what they do is they probably make it into a crystalline solution and then that is just administered as in probably like an IV.
So first of all, you have to crystallize, which is not true. It's true that so when it's crystallized, right? It's in like in the solid state, sort of in the solid state. So that's like lyophilization. Okay, so you can stabilize proteins through lyophilization, which means removing all the water from solution, and then just before administration, you can add solvent back. Right? So that's one way of avoiding cold chain requirements. But most companies don't like. It's a little different with antibodies. Those are high value materials. So you can have even if you have what's called a 2-vial formulation, right? One molecule which is the dried protein, and then another molecule with buffer, and then you mix them. And so because the antibody cost is so much higher and also the amounts are so much larger, it's easy to do all those things. Now in the case of a vaccine, we are not giving 100 milligrams or 1 gram of material, we are giving 20 micrograms. So you can lyophilize the material, but then it becomes very difficult to spot. And then when you add solvent and things like that, you are a little bit uncertain whether everything is resolubilized. The other problem is scale. When you're giving something to 100 people or 1,000 people, then there are many things that you can do. When you're giving things to 1 billion people, you want it to be simple. You just want a vial, take something out, and get jabbed. So there are many, so even for vaccine modalities, it's obviously better, for example, to have what's called mucosal immunization, right? To give it intranasally, for example, because then that goes to the tissues of interest and you get antibodies in the right places and things. But the problem in all of this is delivery. When you give an injection, there's no doubt that the injection has gone in, all the material has gone in. Any other forms of administration, you don't know, right? Whether the full amount has gone in, where it has gone, and also the delivery costs become a little bit higher. So there are many things which are scientifically, and it's known that they are better, but when they have to be scaled up on a mass scale, then that places certain constraints. So certainly crystallization in some instances is possible, but it's not something that's universally applicable, and not every protein can be easily crystallized either. Okay, fine. So anyway, I leave you the answer to the combinatorial problem that I asked was toward getting there, but not quite correct. So you should figure out what the correct answer is.
Thank you so much, sir, for the talk. We would also like to present you with a small token of our appreciation. IGM team leader, and he'll be sharing his experiences with all of us.
I was the IGEM team leader for last year for IIC when there was just one team, by the way. Yeah, and no, one team, right? Okay, so I've called this mRNA, EVs, LNPs, and iGEM because the first three are essentially like what my current fields of interest are. Okay, and how they were all very closely linked to my iGEM experience. So actually, I think exactly 1 year ago, I myself was a part of the All India iGEM meet, as in like as a participant, and we were presenting our project uh, called Metromorpheus to the panel of judges. And what we were trying to do was treat, at least on paper, was to treat a condition called endometriosis, which I'm not sure how many of you have heard about. It's a women's health disorder. We were trying to treat it non-invasively. Okay, now I'm not going to talk about the specifics of the project we did, simply because I've probably talked about it too many times, and this is not the time for it. Okay, but in the end, did it work out in the sense that I had initially expected it to? Not really, but in many more ways than one, it did. So like this was actually targeted at those who were, going to be actually participating in iGEM this time. But what I wanted to ask was, what do you expect from doing this, right? Because no one ever does anything without an expectation of return. You wouldn't do something for free, and you definitely wouldn't spend all of your free time and many hours of your night, which you could have spent on sleep instead, on a science project. Most people wouldn't do that unless you got something really good in return. So if there's anyone who is participating this time and would like to answer that, I'd like to hear it. Is there? Sure. Is there anyone who wants to answer? What do you expect from iGEM? Fine. If there's no one, I'll just continue. Is there? What's going on?
See, what I expect is maybe first, you never get a wet lab experience the first time. So in your first year, getting access to all microbiology equipment and all everything else, that itself is a big thing, and that is one of my main expectation as a freshie. And moving on, it even gives you like an idea how actual research happens, like a small like a small overview. Yeah, and like how to ideate, how to get into the literature review, how everything is done, how actually life is not that simple. So that is one more thing I'd say.
Yeah, thanks so much for that. I was actually not expecting anyone to answer, so thanks a lot. But what did I expect from it? Um, for me, it was just a competition, right? I just wanted to win. And considering that most of us who enter this experience head first, it's like we've just finished our 12th standard, we've just finished our competitive exams. Our first instinct is usually just to compete mindlessly. Okay, so that's what I was, at least. I'm so happy that you're not that, because that kind of outlook is honestly quite bad. But anyway, so because of that, I genuinely lost sight of what iGEM was really about. It was much more about learning than about some sort of mindless rat race. Okay? And I remember, like, after the Jamboree was over, because we didn't get the result that I wanted, even though I had according to me at least, put in a considerable amount of effort. I actually left the Porte de Versailles, where the jamboree was held. I left it like pretty annoyed, and yeah, I just walked off. But uh, that was not the correct response, and it's not the response that anyone who ever participates in this competition should have. Because what we take back from this competition is much more than just a medal or an award or a trophy. Okay, but looking back, what did I get from it, right? The first thing I think was I started to understand why we really do science. Okay, because when we move into uh, a university, for example, most of you freshers, right? If we don't, if we just go ahead, we just go with the flow, you know, we move in with our profs and, you know, start these summer internships and stuff, we generally see research from a purely academic point of view. You know, just about going into the lab, publishing papers, getting citations, and so on and so forth. But we usually lose track of why we're even doing any of this. You know, is what we're doing actually reaching any end consumers? Is it actually having any benefit? And I think because of the specific industrial take that iGEM has, it doesn't have a very academic flavor to it. Because of this particular take, you get to see both sides of the coin, rather than just one, which we would normally see. And because, like you said, you know, this is all about us ideating, us coming up with hypotheses ourselves, rather than just following something that our PI has told us to do, which is what happens in universities. I think even so far as your PhD, from what I can see, right? Because of that, we rarely really understand what we find most interesting. Because a university has very few, doesn't have professors working on everything, right? It has professors working on their fields of interest. And their fields of interest are not necessarily your fields of interest. And it's only when you get to innovate and theorize by yourself that you actually understand what you like. And from our project, I understood that I really liked immunotherapy, protein engineering, and targeted therapeutic delivery, which many people actually don't work on in IISc. What else did I get from it? All of my opportunities and research experiences post that have been a result of my networking during iGEM. Okay, even though I'm not the biggest fan of pure industry or startup culture, and I do like academia quite a bit, even despite that, all of my, like the lab I just worked in for the summer, it wasn't in India, it was in France. I got to know that professor because of my networking during iGEM. Right, she wasn't related to our project, but because I had consulted her and I found out that I really liked her work too, I was able to work in that lab. And I think that experience that I just had has been excellent. And it wouldn't have happened without iGEM. And something else that I learned while doing this, you know, while participating, was also understanding how to manage a team, how to work in a team. Because when we just move out of high school, we usually don't do these things, you know. And more than learning what to do in a team, I learned what not to do, because I made far more mistakes in my management of the team than I did good things. And what was most important, however, according to me, was the feeling of discovery, right? That's not something that we get all the time, okay? And what made this particular experience so much better was not just that we had got a novel result, right? But in our case, at least, for the DNA design, like I had done it, personally. So like I had designed the construct, I'd cultured the cells, and when we finally got a positive result in the end, after making quite a few gambles, because actually in our All India IGEM meet, one of the judges actually said that this design is not going to work in the system of your choice. But turned out it did, right? And I think that also was such a nice feeling, that something that had originated in my mind while I was in the shower, actually, had turned out to work, and I was not expecting it to, right? And it was quite a few months of work that resulted in this. So when we, like, the positive result was actually given by something called horseradish peroxidase. So like it turns blue if the result is positive, if the protein is present. Okay, and I was actually alone in the lab that day, and when I saw the blot paper turn blue, I actually like screamed, and I ran around the lab a few times, because that was by far, even till now, the happiest experience I've had in university, simply because of the joy of discovery. And I think that's something that you would only get if you're doing self-directed research, and the only place where you're going to get to do self-directed research in your undergrad is in IGEM. So this was the final result. It's not blue here, but that's because of the image capturing software. Okay, but this was the most important result in our IGEM cycle. We got a lot more results, but this was the most key one. Okay, and in fact, it was novel in the sense that at the time when we got this result, there was actually just one other industry in India which had also got a similar result with a similar design. So it was actually quite a unique thing. But what did we do after this experience? We did get selected for something called the IGEM Venture Creation Labs, which is where we try to take our idea forward as a startup. And we did make a business model, which the judges in our experience actually liked. It was very extensive. If any of you, I know that there's just one IGEM team here, but if you do need any help with making a business model, you can just mail me or something. You can ask them for my mail or my number, and I can try to help you out. Because ours did actually apparently make sense, even though I didn't think so at the time. Right? But here is one of my criticisms of IGEM. It's that I felt that there was not a very clear way to take it forward. We had, ways to pitch it. We learned how to pitch this thing to like investors and stuff. But for me, it always felt like building castles in the sky. And that's not something I like. Okay? But I thought, maybe it's best to wait for things to take off. Because in the end, fine, you finish your IGEM journey in say like 1 year or 2 years, right? But then you have the rest of your entire education to worry about. And if you really have to do all that with some startup which you're not really sure is even going to take off, and and you're not even given a clear path for it, and it's all very vague and hand wavy, you should probably just wait it out, at least in my opinion. But in the meantime, while I wait for the rest of my degrees, I thought it would be a good idea to actually move forward with at least some of the work that we did. Because I'm not sure how many of you have heard of the condition called endometriosis, but I'm quite confident that it's definitely not all of you, and it's just a few. Okay? So that's why I did start this website. it's actually to raise awareness about the condition. It's not fully fledged yet. I've been saying that for a few months, but I haven't actually had the time to work on it because I've been quite busy. But I will get to it at some point of time, and it'll look better real soon. But I think at least this is my way of making a difference for now. And none of this, none of my experiences, none of my research or outreach experiences would have ever happened without AIIM. So in summary, right, like I said, be clear what you want from the experience and make sure that you just don't think it's some sort of rat race. Research is much more than that. And it was because of my AIIM experience that I learned that. Okay? And the second point is actually also very important because I've seen many teams do this, but they usually switch tracks or move to easier tracks because it's the easy thing to do and there's less competition there. What I'd recommend is that this shouldn't be done because it's not common that you choose the field, right? The field chooses you. When you first enter science, you're not usually going in there saying that I want to be an immunologist or I want to work with T cells. You're usually going there in there as a blank slate. And the first few people that write things on it are the things that stick. Okay? And if you start off with something mundane and bland that you don't really connect with, then that's actually a bad start. So maybe you should worry a bit less about the prize you're trying to win and worry more about how this is actually going to affect you later on. Because, see, look, I wouldn't like, for example, working with mass producing some protein in E. coli bacteria. I wouldn't like that at all. And I would never move into that field of research. But if we had tried to move on to something that was easier to do or easier to do well in, then I would have been stuck with a mundane project that would have never led me on to the experiences I've had now in other labs beyond that. And the reason you should do that, of course, is because you need to work on this for a considerable period of time and give up a lot of your so-called social life and so on. Yeah, and finally, this is my opinion. As I always say, you guys should make your own opinions. And they should be very well considered. Okay? Thanks, and all the best.
afternoon and welcome. So I understand some folks went out, not sure whether to join. So next few minutes I'll try to motivate about the work. It's just a small exercise that I did during the COVID times, and my effort here is to highlight, how computing can help. At times, when experiments and lots of different observations are not consistent with each other. Okay. This work was done during the COVID times. So if you're interested in the paper, so this paper was published in 2021, and questions that we asked is, why is SARS-CoV-2 so contagious? Right? During those days, everybody was trying to come up with a theory that what is this new virus? Why is it so contagious, right? So you had a number called R0 that was being bandied around, and each of the different R0 values of different viruses were being compared. But the SARS-CoV-2 R0 was very high. The infection was spreading rapidly. And many people, from different backgrounds jumped into the fray trying to explain that what is this unique virus that, causing so much havoc through infection. So the key question is, why is SARS-CoV-2 so contagious? I'll explain the background of the work. What motivated me in trying to do this work, because we had some previous experience working with beta coronaviruses. So SARS-CoV-2 is a beta coronavirus. So we had some previous experiments, and then the current results we got at that time, and this led to me to propose a infection initiation model, which I hope some experimentalists will prove in times to come. So when we look at the different kinds of theories that went around in those times, there was one proposition that came out, that why is SARS-CoV-2 infecting others? So one person wrote a letter, wrote an article saying that the virus lives very long. 72 hours is its lifetime. So because it lives long, it infects more. So that was bandied around, and he did a aerosol and surface stability comparison with SARS-CoV-1, and he published this paper. Next, the person 2, okay, he came with a proposition that person's coughing can produce long plume of aerosols, and this can lead to infection, especially in indoors. That was the second proposition. But in each of these propositions, should have been valid for the other viruses as well, right? Why just SARS-CoV-2? In fact, beta coronaviruses have the largest genome among the known viruses, 30 kb, 30 kb, yes, I think. It's one of the largest genomes that are available, and its particle is quite big, right? So it did not make good sense when I read these articles. Another person, just opposite to what the previous report said, says the aerosols that are formed by these particles are very small, so they can, when they get into our nose, they can go deep inside and infect our lung directly. Okay, but if we saw the infection pattern, SARS-CoV-2 always infected the upper, respiratory tract, and slowly then it went inside. So also this did not gel well with what was known. So then there were other propositions, and these were molecular propositions. So to understand those propositions, we'll need to understand the virus a little bit. So coronavirus is the name because the spike protein that is shown here. So that is on the surface of the virus, and these spike proteins give it as a corona-like, image. So that is why it's called the coronavirus. And then there are important proteins like the envelope protein and the membrane glycoprotein, the spike protein that are important and that are on the surface, right? And things to note is that this molecule here is quite big, right? So it spans about 150 angstrom in width at its head, and this is about 75 angstrom. So these are quite big, molecules. Okay, and they're on the surface. So, molecular biologists, structural biologists, they came up with, a proposition that the surface of the spike protein, has been made hydrophobic because of a lot of, glycation. Okay, so this spike glycoprotein that is eventually formed has quite a bit of hydrophobicity on the side, on the surface, that is allowing it to, become sticky, and wherever it gets stuck, it is causing increase in infection. So this was researched, and they said there are 23 sites that are glycated. But you won't believe what is the size of this protein. So this is a trimeric protein, and it has total length of the protein is about 1350 amino acids. So this is such a large protein, and of that 23 sites in each of the monomers were infected. So although on picture, they were made to look very large and covering the entire surface, in reality, they were a very small fraction of the entire molecule. So again, we thought that, it doesn't really make much of a sense, right? So then another proposition was the high binding affinity to the ACE2 receptor. So the spike protein, when it recognizes the host protein, the recognition happens to this receptor protein called ACE2. So somebody said, this spike protein is having a higher affinity to this ACE protein, so that is why the infection is more. But if we did a molecular, calculation, because these cannot be really done experimentally, right? So the molecular calculation based on different kinds of, affinity based evaluations did not indicate a, dramatic change in the affinity values, right? So although some papers, tried to propose this, it didn't reveal that the R0 value, so infectivity value, would have been so dramatic, because the affinity was not too different. There was a moderate, very small difference in the affinity. The next proposition came that the spike protein was very efficiently primed. Okay, so now for those of you who know how these infections work, these are the theory, and not that the virus infects only through one of the mechanisms. There are several mechanisms. So one of the mechanisms said that there are two sites of cleavage of this protein, and one site is already cleaved, so that the protein is ready to infect when the other sites get cleaved. So that is what is being called as priming. So they came up with a theory. Okay, I don't have that slide. So they came up with that theory that priming is helping the protein to come up with efficient way of entry. So with another line of thought, which I also, I was thinking at that time that why is it? So I thought that the virus replicates very well inside the host. So while a lot of virus shedding is going on, from the infected person, maybe that is responsible. But again, this just was a thought from my side. So these were the different propositions that were available at that time. And as you can see that nobody was satisfied with the others' proposition, so that the new propositions kept on coming, right? So after this sixth proposition, then I started to think that no, there must be something different. So if you have to analyze, we have to look at the pathway. So there are two kinds of pathways that are normally used by the virus for entry. One is the direct pathway. Here is, you can see one, right? So one gets into 2B and 2A. One is the direct pathway, and the other one is the endosomal pathway, where it penetrates inside and then it gets in. So these are the two available pathways. And that previous paper that I showed you, a proposition when the virus is primed, it said that there are two of these cleavage sites, and due to this priming, one is cut, the other is ready to launch, so other gets cut, so immediately the infection happens. Okay. So it said that it is activated spike protein that makes it very infectious. Now we were trying to see whether these made sense or not. Okay. So previously we had worked with the mouse hepatitis virus. The mouse hepatitis virus is also a beta coronavirus. It's a very infectious virus, and it also is so virulent. If it infects the liver of the mouse, it penetrates and it even goes to the brain, and it creates meningitis and destroys the cells, astrocytes, causes astroglioma and things like that. So we had previous knowledge about this virus because I was, even before this COVID happened, we had published some papers. So we tried to draw our inference or rationale or logic from that. So if we look at the cleavage sites of the different kinds of spike proteins, you will see that these are the cleavage sites, and you can see that cleavage site has R. So if you have R, then it can cleave. But you will also find that the SARS-CoV-1 does not have a cleavage site. Okay, so then how is it infecting? Right? If that is essential, then that should also have a cleavage site. So this said that no, it's not the only way for entry. Right? Then there was talk about glycosylation. Right? We were only looking at molecular aspect. So the aerosol versions of those were not covered in our analysis. Next is glycosylation. Now if we look at the glycosylation, so you will find that for a spike protein to be glycosylated, you need this signal, KH, KIHIK. But if you look at the MHV protein, which is what we had previously worked with and knew, it did not have any glycosylation signal, neither does it get glycosylated, which ruled out that glycosylation cannot be an essential property that will increase or make the R0 value so high. So we ruled that out. So the real question then, what is it that is causing this high infection? Okay. So then we were scratching our head. Okay. So we thought, and this was a question that was posed by my collaborator who had worked with the protein. Now during infection, the fusion peptides are believed to be the early mediators of contact, which means when the protein, the spike protein, the virus comes, it attaches to the receptor, the host cell receptor, and then as soon as it gets stuck, when it falls on the host and then penetrates inside, the fusion peptides are believed to be the first points of contact during this infection. So that is why these are called fusion peptides. Okay. So the sheep said that there must be something in the fusion peptide. So then I started looking at it. So fusion peptides or spike protein have a key role to play in virus fusogenicity and consequent infection. So that was the hypothesis. So now to do this analysis, of course you need a good understanding of the structure of the spike protein. So the spike protein, you can see it's a trimer, as I told you. The head looks like a triangle, okay, and the MHV one is 140 angstrom kind of long, and this is about 140 angstrom wide. Now in this MHV, spike cleavage is not mandatory, and spike also does not have the canonical S1 S2 cleavage site, and it has limited glycosylation. Okay, so that is telling that the infection is not contingent upon those properties. It really rules them out. So then we went and checked, there are other infectious viruses also. We have the Ebola virus, what is this, avian sarcoma leucosis virus, and there are other viruses. I think I have it in the other slide. Ebola virus. These are also very infectious, viruses. So we went and tried to look at their fusion peptides. What is the property? What we found is that their fusion peptides had prolines in them. They had a single amino acid proline in them. So these were in literature called as central prolines. So MHVA59 also had proline in them. So this was a line that you have a proline in there, so somehow it is helping in the infection. So what she did, okay, she introduced MHVA59 had one proline. Okay, so two prolines in the wild type, and she deleted one proline. When she deleted one proline, the fusogenicity of the virus came down dramatically, which means the infection was getting impaired. So there was something in the fusion peptide that was causing it, right? And it was believed to be the early mediator of infection. So now this work was already done before the COVID. So that was these experiments were giving something. Okay. So then what is proline? If you look at it as an amino acid. The basic fact is that the proline is not an amino acid, it's an amino acid. And because it's an amino acid, the Ramachandran plot, it doesn't cover all the quadrants, right? So this quadrant is prohibited, and most of the locations that the phi psi angle populates is restricted to these three regions. So existence of a proline makes that segment very rigid. So the peptide is floppy, but you put an proline in that region, it will make it rigid. Okay, so there's something got to do with the rigidity. Okay. So we did some molecular dynamic studies. Okay. These studies were done previous to the COVID, and we clearly found that if we had a single proline and double proline, these experiments were done only on the S2 domain. The spike protein has two domains, S1 domain and the S2 domain. The S1 domain mediates the receptor binding. The S2 domain mediates the fusion. So we took the S2 domain and we found that yes, when we just take the S2 domain and proline is there, you can see that whenever proline is there, let us see which one is which one. It's kind of not labeled, but definitely the one that is more rigid would be the one where there is double proline, and the one where it is not, it will be single proline. So we did more investigations and we created mimetic peptides where we took these peptides and we did NMR. So when we did NMR, we found that the cis, when we had a proline, it also led to cis-trans isomerization. So when you had cis-trans isomerization, which means that it could also, you know, the hydrophobic side can invert and come out, right? So having an extra proline and cis-trans isomerization allow the peptide to flip. So these are kind of properties that gets imbibed in the the fusion peptide. So then we tried to find out where are the fusion peptides of the different spike proteins. So we did a long list of alignments. And what we found that in the literature, there was no agreement on which are the sites of fusion peptides. Okay, so paper 1 said the fusion peptide is here, paper 2 said fusion peptide is here. And after a lot of confusion, I figured out that since there is no direct experimental evidence, these are calculated from a hydrophobicity scale. Okay. So we also did the same calculation using the hydrophobicity scale, and we figured out that there are 3 prospective fusion peptide sites in the SARS-CoV-2. Okay. So that's what we figured out. And when we went back, we thought that we will now categorize or kind of structurally computationally investigate the characteristics of each of these 3 fusion peptides. So then we mapped those fusion peptides regions on the surface of the protein, and we found out that this is present in this region of the protein, and this is where the virus is. Okay. Yes, yes. So we found that these were located here. The receptor binding is somewhere here. The fusion peptides are here, and the virus is here. Okay. So you can see that the host cell is here, the fusion peptide is here, the virus is here. So it has to go inside because this is most proximal to the host. The virus has to first, then it has to like a spear penetrate, but the fusion peptides are present in the lower base. So what is the conundrum here? Okay. So these are some of the pictures that we created using electrostatic potential calculation, and we also did molecular dynamic simulation, trying to figure out which peptide is rigid, which peptide is flexible, and we mapped them. Okay, so we can see that when we look at, so let's see what the graphs are. So these are all RMS graphs. Okay, so what we see here, so we have a proline here. So as soon as you have a proline here, you will see that this will dip. Okay, whenever you have a proline, you will see a dip. So there were some other cases where there were no prolines, and when we compared, so these regions were equivalent to other viruses. Now we had to explain the SARS-CoV virus, right? Now whether SARS-CoV also had a proline in that region or not. So when we looked at this, so where is SARS-CoV-2? Let's see. This is SARS-CoV-2. This is FP1. This is FP1, fusion peptide 1, fusion peptide 2, fusion peptide 3, and this is SARS-CoV-2. When we look at the fusion peptide here, for which we had done an equivalent experiment in MHV, it was the fusion peptide 3 region which was being compared. So they had the proline. So this is the proline that it had. But in this case, there were no prolines, right? So then, what is it that was creating the difference? So then we figured out that there is a very steep dip here in the flexibility. And this is because there is a small set of residues here called TIT. Now for those of you who know about amino acids, T, I and T are beta branched amino acids. You know amino acids have long chains, some are single, they're just simply straight. So isoleucine and threonine, they are beta branched. So what happens is, as soon as the side chain gets branched near the main chain, it clashes with the main chain and it cannot rotate. So instead of a proline, very likely that this was creating a similar effect like a proline. So then this was the explanation that we found, and then we did the regular dynamics, and we figured out that it was indeed creating a restriction. Just take 2 more minutes since I'm out of time. So now the question is that the fusion peptides were all on the back side, right? The virus was here and the receptor binding domain was here. So in this picture I am showing you that if it has to hit like a spear and get inside, then it must be anchored in a tripod, right? If you have to stand and pins inside, you have to stand on 3, right? If you are only attached to 2 receptor sites, then also you will fall, right? If you have to push yourself in, then you must stand in a tripod, because mechanics says that you need at least 3 attachments for having a pincer inside. With 2 you cannot, and in 1 of course you cannot. So now what is happening, that if you look at the surface of the virus, the presence of the receptor protein has to match the exact presence location of the receptor the spike protein receptor binding domain, right? Then only it will bind. Now what is the probability that you will have exact 3 receptor sites matching the virus receptor binding domain? Probability is very low, right? The maximum probability is that it will bind only 1. So now if it binds only 1, then what will happen is that when it falls, it will fall on its belly. So that is what I call a belly landing. And if it falls, then it will fall on the fusion peptide. That will be the first place it will hit, because of the shape of the protein. And to explain this further, I created a small model. So you can see the front side, the top side of the head is like a triangle. So if the triangle falls on the surface, it will always be on the side. And if it is so, the fusion peptides are shown in green here. As soon as it falls, these are the locations that it will touch, right? Structurally it makes sense. So then it falls, it first hits the fusion peptide, and when it hits the fusion peptide, then it starts penetrating, and while the structure conformational change is happening, it cuts through the membrane and it gets inside. So this is what we found, and I was very excited that we found something that is amazing, how the entire spike protein architecture is evolved. So this was what we concluded. Spike proteins are and diverse, and finding common features is key to understanding their structure and function. Fusion peptides are key fragments strategically located for protein-host membrane interaction. Rigidity of the fusion peptides enables it to perform efficiently in engaging the host membrane and its disruption. electrostatic, other physicochemical properties are also important. Not that they're not important, they're important. Given their importance, fusion peptide surfaces are legitimate targets for therapeutic intervention, and they can be considered an important factor in SARS-CoV-2 spread. Subsequently, we have also done experiments using fusion peptide mimetics, and we have a paper under review right now, which hopefully will be accepted, where we have shown that these, if you can block these regions, then you reduce the infection. So you can see, just by computing, looking at the structure, combined with experiment, you can come up with nice explanation and design of experiment. With that, I'd like to acknowledge the collaborators. There are several students involved in this work who did the bulk of the heavy lifting. Thank you so much.
Are there any questions?
The fusion peptide signals, like the TIT signals in all the spike proteins.
Very surprisingly, if you look at the virus sequences, the most mutated domain is the S1 domain, whereas the S2 domain is highly conserved. In fact, if you look at the many different families of the beta coronavirus, these regions are not changed at all, which also lends to the credibility of what we are saying. These are very important for its infection.
Are there any more questions?
Thank you very much for the talk, Professor. I was just wondering, so I'm not sure when this was published, like
2021.
Yeah, so were there any attempts to make vaccines towards more conserved parts of the spike?
So unfortunately, people thought that if you can disrupt the receptor binding domain, that is where the mitigation lies. So this is the first time that we are trying to base a design on the S2 domain. And as I said, that the paper is under review currently, where we have shown that mimetic peptides for the fusion, peptide can also reduce the viral infection. So if this paper comes out, then now people will also try to design a vaccine based on this.
That's great. Thank you very much.
Anything else? Thank you so much, Dr. Detna. We would also like to present you with a small token of our appreciation.
Hello. Hi, Varsha, can you hear me?
Yeah, I can hear you.
Okay, so we're happy to begin today's fireside chat with our IGEM alumni and IGEM community alumni as well. So, to begin with, Varsha is an IGEM competition and community alumni. She was a member of IGEM ISER Pune in 2019. She was an IGEM judge in 2023. She was a mentor to VIT Vellore in 23. And she was also the IGEM ambassador for Asia in 2020, the IGEM community coordinator in 2021. So you have a host of experiences with IGEM, and I'm so glad that you could join us today. We're so glad you could join us. Would you like to tell us more about your experience with IGEM after the competition and the various positions that you've held since then?
Right, yeah. So I think I finished I was part of the competition back in 2019. And after that, I really loved my iGEM experience. I loved interacting with all these people from around the world. So that's what, so I decided, I want to continue staying involved with iGEM. So I applied which was for iGEM, what is called iGEM community now, but was called after iGEM back then. And yeah, I became an ambassador to Asia. I have served as judge. I have served as mentors for a lot of teams. Um, And the thing about being part of iGEM community is that you can network, and there's nothing more powerful than networking for your career. You can meet professors from around the world, right? You can immediately reach out, send them an email, and if it says I was part of iGEM, that statement itself carries a lot of power. And ideally, professors would have, they would have ignored your emails or they would have not looked at it. But having that one statement in your email or your LinkedIn message is what really makes people reply to you. So I would say my iGEM experience was very fun. It was very powerful because it really helped me build and shape my career. And of course, I also have a lot of good friends in iGEM now that I hang out with regularly. So yeah, fun and powerful.
Okay, so can you tell us about the various possibilities with iGEM community that people who can get into after the competition?
Right. So now there, with iGEM community, there are like some formal opportunities that the iGEM community itself puts out. And then there's a lot of informal networking opportunities that you really should take note of and make use of. One is you can actually undertake a project, right? So you can put out an appeal for a project with the iGEM community. And if there are people who are interested in the project, they'll join you. So that's like build that actually helps build your CV, because when you look at your undergrad or your master's experiences, you really don't have anything to put in your CV apart from maybe the courses you've taken. There's really not much to put in your CV. And one really good thing that you can put is project experience. So if you and these projects really help you build all kinds of soft skills, which is generally what employers or universities look for. So not only do they care if you can run a molecular assay in lab, or if you can do a computational program or a model, but they also care if you have skills like communication, project management, expertise like that, right? And all these soft skills are what really matter. And this project, like doing a project with the iGEM community, is a very good opportunity for you guys to build those skills and show it on your CV, to have something actually concrete that you can show on your CV. So that is one. The second is networking. Like, you really should make use of the network. Everybody requires a mentor, right? So reach out to people on LinkedIn, reach out to people on the alumni network, reach out to people. You can email professors who you can see have been part of iGEM projects, right? And just say, hey, can I have a chat with you? I'm interested in your career. And if you use that word that I was part of iGEM, and I heard of you through iGEM or something like that, people are more than happy to mentor you, help you out. Anything. It could be even, if you're applying for a PhD and you need somebody to now, go through your application once, give you tips. It could be anything like that. It could be that. It could be what career opportunities you can pursue, what research you can pursue, anything. So always seek out a mentor within the network, I would say. Seek out a mentor and seek out other connections. So ping a random university you're interested in, ping people in the industry you're interested in. There are a lot of people in biotech, pharma, that you would be interested in. And networking is very powerful. It's mostly how people get jobs. And the third, I would say, is you actually, so in your undergrad, if you look at how a lot of Indian schools are taught, you don't really have the opportunity to explore beyond your course. You only mostly study about your course in textbooks, which is very different from actually working on something hands on, and very different from the experience of exploring something hands on. So I would say use iGEM community to explore things like science communication, policy, and aspects like that that you would not cover in your basic lab courses. Yeah.
Okay, so you're currently pursuing your master's in control of infectious diseases from the London School of Hygiene and Tropical Medicine. So, do you feel like in any way that iGEM community has helped you shape or make that decision, or any of your positions in iGEM really?
Yeah, so my previous master's was mostly in immunology, and I was very lab focused, right? But then because of iGEM, I actually ended up with a lot of lab experience and actually working in labs. And I realized that if I spend one more hour in a four-wall lab with the centrifuge running in the background, I will go mad. So while I thought immunology was very fun in my classes and coursework, I realized actually working on it hands on, it was really boring and the lifestyle was not the one I wanted. And that is mostly because of iGEM. Otherwise, if I would not have realized this, I would have probably gone on to do a PhD in it, and I would have suffered my way through it. So I decided that I had enough of lab, and because I worked with iGEM, I was doing the iGEM coordinator position. It made me realize that my strengths are really more in like project management, um, and that's what I really enjoy doing. Like I enjoy working with people, I enjoy solving problems in a more fast-paced world. So that's why I decided to move from immunology, the scientific part of the immunology, to more of disease control programs and more of translational medicine, which is what I really enjoy doing. So that was making me switch fields.
Okay, thank you, Varsha. I can open the floor to questions from the audience.
Yeah, are there any questions from the audience?
About joining iGEM community, why you should join, what are the options available. If you have any questions, do let us know.
Hello, am I
Yeah.
visible right now?
No. Oh yeah.
Yes. So, how did you exactly transition from being just an iGEM member to being such an important person in the community?
Yeah, so I am an extrovert, so networking is easy to me. I understand it's not as easy for a lot of other people. So what I started is I started talking to the ambassadors who were already part of the IGEM community, and I became really good friends with them as well. And they told me a lot about the opportunities. Otherwise, you can still look out for the opportunities IGEM puts out on their IGEM channel. You can go to their webpage, the IGEM community webpage, and look at the opportunities, but that is always harder to find. So the best way to do it is to actually talk to ambassadors and ask them about what are the opportunities there, to keep constantly following up with them, right? And they will let you know of what really the opportunities are. And when those opportunities open up, I would suggest you apply for them. Or you can even come up with something of your own, like take your own initiative, say I want to do this, and go to one of the ambassadors, go to one of the coordinators and say I want to do this, and they will most likely support you in it.
I see. Thank you.
Are there any other questions from the audience?
Okay, thank you so much, Varsha, for joining us. We will conclude the session for today.
No worries, good luck and all the best for all your projects.
Thank
Thank
you.
you. We'll be serving tea and coffee. Hello, yeah. We'll be serving tea and coffee outside in a few minutes. After that, starting 5 o'clock, we have the closing session. We will have the results of the judging sessions announced there as well.
Hello. Yeah, it's the tea break, so please have some tea and biscuits. And please come back by 5.
doing this almost 15 years back. I know him so well. He was keep on telling, because I also do biology. Whether I say synthetic biology or just biology, although I'm a physicist. So, but the major part of my work is actually biology, computational biology. So Mukund, I know him over years, and he used to tell earlier that he's doing synthetic biology. So that's what I'm trying to figure out, this computation, what it is actually. That's what I'm trying to figure out. Because I already thought it's quite a mature field.
So actually, in one of our talks, we had an IBM alumnus speaking who went on to develop a startup from that project. It is called T-Safe Energy. So she was telling the students about that. And
Where is that startup?
it's based in Bangalore.
Bangalore?
They're harvesting
She was alumni?
No, no, no.
He's from IIT Madras.
IIT Madras. She was one of the insidious alumnus. He was doing his PhD with Sachi, Sachi Goswami. Did my course also. I think he had also a startup on similar lines. I'm forgetting what is his name. He's also from IIT. He did his research from IIT. PhD with Sachi Goswami. Sachi Goswami, you know? She's also in insidious. He has also started along the similar line, quite several years. Or you are working in a... Okay, good.
Good evening, everyone. So finally, we have reached the closing session of All India IGM Meet 2024, organized by ISC. So for those who don't remember me, I am Shubhanand Banerjee, the team leader of ISC Bangalore team. So not taking any more time, I'd like to invite our Dean, Professor Prabal Maiti, to give his precious words to the audience. Sir, please.
Thanks, Shubham. And I'm happy to hear and see you all. I have heard about this ISAM events a few years, but really did not look at the details. Now I am looking at the details. That's what I was asking so many questions. So as I gather, it's a lot of excitement, lot of new things, lot of expense also I see, because many of the bills I had to sign. So that's actually my first encounter actually. So I'm happy to see that several other participants from IIT Chennai and IIT Mumbai and many other ambassadors are here. And so I hope that like previous year, you bring laurels to country, IISc, and do well again with fantastic development or ideas basically. So if I got it well, is the idea which has to be translated into something tangible. So I'm looking forward to see the October, correct? October October event in Paris, and hope again it will be a really fantastic news for all of us. So wish you all the best. Okay, thank you.
Thank you, Professor. So now I'd like to request the ambassadors to kindly announce the results of the judging sessions.
Hi, everybody. After a grueling 3-day conference, we finally um, have the judges' verdict. So, earlier in the morning, we were looking at the scores of all the teams. We had to tabulate a lot because there were so many metrics that all the teams were they were judged on. And as you mentioned earlier, we have 5 different recognitions: best overall, and then a winner for each criteria that was mentioned, which is best impact, presentation, engineering, and human practices, in that order. The judges had a very, very nice time looking at your presentations, and it was really difficult to judge on the basis of all the metrics, because we were trying to emulate the metrics seen at the Jamboree. And we had 5 different judges this time. They are not here with us today, but 5 different judges, which means that your scores are all very carefully decided, because there are 5 different people who are making the decisions for the teams. Okay, without any further ado, I think we can announce the recognitions. We'll start off with the last with criteria 4, best human practices. Any guesses? I'm sorry for doing this, but I have to do this. Good guess. Any other guesses? Okay. No other guesses. Best human practices, MIT Mahe.
So for the next category, which is best engineering, we have ISIL TVM.
Best presentation. Any guesses? Best presentation. Okay. IAC UG.
Okay, for the next category, best impact, we have ISCUG.
Okay, so now I'd like to invite the Human Practices Coordinator, Parth Kumar, to give the vote of thanks to the audience.
Thank you, Shubhanan. So, we had a lot of teams joining us from all over India this year for the All India IGEM meet. Thank you so much to all the teams who could make it here on campus, and of course the two teams who joined us online, IISER Kolkata and IIT Roorkee. So we had IIT Bombay, IIT Madras, IISER Kolkata, sorry, they were online, IISER TVM, MIT Mahe, REC Chennai, SVC Chennai, VIT Vellore, and the two ISC teams, as well as RVCE, which will be RVC Bangalore, which will be participating in IGEM starting next year. They also came to spectate, and I hope all the teams had a good time, and we certainly did have a good time hosting you, and we're very glad to have been given the opportunity to host all the Indian IGEM teams for like a mini jamboree, giving them the experience to collaborate, talk to different people, and of course have the perspective of future collaborations with amongst each other and with the judges and other speakers as well. So yeah, now I'd like to deliver the vote of thanks. So from our batch, from our team, our ISC IGEM team, we had Shubhanan, the team leader, Suvam, the co-team leader, Avni, the wet lab coordinator, Anurag, the dry lab coordinator, Geet and Anushka helped with web dev and design, as well as Ashik. And Manya and Harish were also there with us at every step of the process. Kabirdeep was there helping us stream the event. He's also a part of the human practices team. And from the ISC software team, we have Trishna, Anirudh, and Sushmit. Our seniors also, given their much greater experience in the process than us, they were there with us throughout the process, helping us host the event and also in IGEM. So special thanks to Kush, Kartikesh, Lok, Dhruv, our senior Aditya, the last year's team leader, Rahul Bhaiya, of course, and Adarsh and Shekhar. Then our juniors also who recently came to campus, they volunteered and helped us out with the event a lot. So I'd like to take their names as well. We have Arav, Viraj, Durga, Hasini, Mihir, Sukhbir, Abhay, Aditya, Mukta, Chidambar, Devansh, Jashandeep, and Ritubroto. Then I'd also like to extend a very heartfelt gratitude to our professors who came to speak in the event. First of all, we have our primary PI, Dr. Deepak Nair. We also had participation, as you saw, from a lot of speakers from a lot of different departments in ISC. We had Professor Devnath Pal, Professor Atri Ghosh, Dr. Sanhita Sinhare, Dr. Ramrai Bhatt, Professor Shashi Thutupalli from NCBS joined us as well. And of course, we had the IGEM team's mentors, Dr. Raghavan Varadrajan and Dr. Ravi Mudashetty. I'd also like to extend my gratitude towards the speakers we had from outside of ISC and the judges as well. On the judging panel, we had Dr. Lavanya from ISA Bhopal, Dr. Selvi from ISA Berhampur, and on the online judging panel, we had Mr. Onkar Date, Dr. Preeti Srivastav, and Ms. Shruti Sridhar. As well as for speakers, we had the IGEM ambassadors had invited Ms. Sailaja from C6 Energy, and we had Ms. Varsha as an IGEM alumna. Also, most important of all, I'd like to extend my gratitude to the administrative people in ISC as well as other people who helped us out at every stage of the process. Of course, we have our Dean UG, Professor Praval Mehti. We have the Associate Dean, Professor Shrikant Iyer. Our former Dean, Professor Balaji Jagirdar. We have Mr. Ashwath from the who's the section officer in the UG department, and Ms. Haripriya from the UG office. Of course, we'd like to extend our heartfelt gratitude to help us host the event here to the Registrar Captain Sridhar Warrior, also to Dr. P.S. Anil Kumar, who's the Dean ANF, for his support throughout the process, and to Mr. Joydeep Deb from the JNCSR, because our judges were hosted by them in the Jawahar Guest House, to the Joint Registrars of Finance, of course, the Office of Communications and the Department of Developmental Biology and Genetics. Also, we would like to thank the ECE department and IEEE Connect for providing us with the venue to host the event. And last of all, I'd like to extend heartfelt gratitude to the director of ISC, and without whom this process would not have been possible at all. The IGEM ambassadors, Srimati, Hana, Sasha, and Muskan could unfortunately not join us here. Thank you so much to everyone for joining us for AIIM 2024. We hope you had a good time, and we'd love to hear back from you and love to collaborate with you in the future as well. So yeah, with that, we conclude the All India IGEM meet. We'll be meeting for a group photograph in front of the main building soon afterwards. Thank you.